I'm going to talk about something that is certainly controversial always, that is the notion of resolution and quality, and what can you do with that. So I'm going to talk about directional resolution, how we are using that to understand better the quality of a map, among other things, and I will also talk about how the notion of local resolution, considering that as an additional information, is helping us in getting better maps through a deconvolution process. So basically, the notion of local resolution has been fundamental in electron microscopy. Really, we have one of the powers of our approach is that we can deal with flexible structures, and flexibility is going to be found in different parts of the structure. Normally, here you have TRPV1, and the center, the transmembrane part, is better ordered than areas that are farther away from the membrane, and this is something that, in principle, is common. The local resolution concept were introduced in the area by these two papers, Blockres and ResMap, and we followed with MonoRes, that is based on the use of another type of signals, monogenic signals, but at the end we are doing a signal-to-noise ratio between the power of the monogenic signal and the noise. But essentially, this is the state of the art, if you want, in terms of local resolution. What have we done more? Well, we have thought about the following. Listen, it is fantastic if I give you the resolution in one of your voxels. But what about if I am able to tell you how you are doing in that particular voxel along different directions? So not only to tell you that you have 7 Armstrong resolution, but actually that you have very good data coming from that direction and not so much good data coming from that other direction. So what we did was to modify MonoRes in a way that we were working along direction, a cone of directions. The center of the cone, we took it as the direction we were working with, and for the rest, we used the normal algorithm that we had used before to calculate the local resolution. So it was just to filter by the frequencies within that cone and to ask how things were. Okay, there is. Okay, we can, if we have that notion of directionality in the local resolution, we can ask many questions. We can really, if I am in the center of alpha helix, I am expected to have an important component, an important frequency that would be related to the radius of this alpha helix, or perhaps to the length of this alpha helix in another direction. But let me talk about one of the multiple applications, which is the tangential resolution. And I say one of the many multiple applications because I think that this idea of directional local resolution could be used in many problems, and could be a way to solve a number of them. What did I do here? Well, here you have the same slice from a map, and what I have done is just to rotate one of them. And then I have added and I have obtained a structure that is obvious that since I have introduced here a rotation, I have not corrected for that. I'm going to have a central part that is more well-defined, and then a part that is more blurred. And using very simple geometry, I can say, well, listen, if this is my structure and I have the center of rotation in the center of my structure, if I have a certain angular error, well, the angular error is going to create a blur, is going to create some uncertainty of where this point is going to be. And this is actually an arch. So if I use the very simple expression of arch is equal to the angle multiplied by the radius for small angles, well, then if I plot as a function of radius, I will have immediately information about my angular error. Okay. How we test it with computer simulated data, like everybody here, error-free. Well, error-free, just I'm going to talk only about the radial resolution, so, or excuse me, about the tangential resolution, so I will not talk about other curves. What do we see when we have error-free data? We see that really, as we plot the resolution, the tangential resolution over radius, well, we don't see any slope. Well, since we didn't have any angular error, we don't have any slope. Now we introduce an angular error, and we see that really these curves, I mean, we see a curve with a certain slope. These divergences here are, this is beta-galactosidase, and they are related to being, when I start having more or less pixels that are occupied by areas outside the protein. I mean, it's not a spherical specimen. And what happened with translational errors? Well, with translational error, nothing I have told you about tangential resolution is related to translation. It's related to rotations. So if I make errors in translations, I get a worse resolution. Fine, I get a worse resolution, but I don't get any change in actually the slope of the curve, and I have a flat curve, as it is logical having a noise-free data from the point of view of angular error. Okay, so this is what we should obtain, and this is what we are obtaining. And now we apply to a number of cases. Let me present it, the proteasome and the ribosome. In the case of the proteasome, we see that really there is a small slope in that curve, but really is quite flat. So in general, the map has, on average, a small angular error. And understand what I'm saying. I'm saying that I take the map. I do not take the images. I do not have the angular assignment of these images. And only from the map alone, I am telling you something about something as important as the average angular error that you are doing. So this is an important information related to the quality of your reconstruction. Well, we also, if you want, an opposite case was actually the case of this specific ribosome that was used before by Chris Russo in another paper, and we just wanted to use the same one. And really, the conclusion was the same that he reached, that in terms of angular errors, this map has more angular errors than the other one. The difference is that we, again, didn't have the images or the angular assignment of the images, only the map. Okay, we did something more with this angular or this directional resolution. This plot, you are certainly very used to them in Cryo-EM. Normally, they refer to the number of particles that this is the sphere of projection directions, and here normally you present the number of images that are coming from this projection direction. Okay, so this would be the normal way to present it. Here, I'm not presenting that. What I'm presenting is actually the number of lines that go through a pixel along this direction that have the maximum resolution, or normally we try to work always with the 5% highest resolution. And what we are seeing is that there are areas here that most of the lines that go through pixel along that particular direction, they have almost all of them the maximum resolution. While we are finding that there are other directions in which the spread of resolution is very large. What is that telling us? Well, this is telling us a number of things. First, maybe really there are less images in that particular direction, but more importantly, possibly for that particular direction, I have more problems in assigning the angle, the correct angles. I mean, the problem of angular assignment is complex and depends on the shape of your object. And there are certain positions from which that shape is more difficult to see small changes than from other positions. And what I'm saying is that from direction like that, I have a large spread of values of the maximum resolution along that direction, and this is telling me that I have a spread. So I probably am not determining very well the angles of these particles. It could also be telling me that there is flexibility, and that flexibility is particularly noticeable from that direction. Fine, yes. And it would be unable to separate one from the other. But this would be an important piece of information that is not as easy as just counting the images, but is more related to actually the shape of your particle, how easy it has been, how successful has been the process of assigning angles and things like that. So this is the contribution to the map validation, to the establishment of the quality of a map. And this is one application, this is one GPCR reconstruction from Saxton group in Monash, and you see that really it was, we were working with him to see how well determined the angular assignment had been in his case, and really it was a very good example of very good determination of the angular assignment. So we were happy in that we had something new to contribute to the field of validation. But okay, besides being able to tell you that the map is good or not so good, am I able to use this local resolution to do something good for the map, to get a better map? Well, yes, we are in a simple manner, conceptually simple manner. This we have not yet done. We are very close to do it, but we have not yet done, including directionality. So what you are going to see is in an isotropic manner. I'm not considering the directionality, but we will do it very soon. Well, the idea is that virtually any system is going to introduce errors, and you have a perfect image, and this perfect image is going to appear quite imperfect depending on the errors that the instrument introduced. This is the case of the Hubble instrument, and through a deblurring process you get, or they got, normally quite good images. In the case of the Hubble, and in general, we are able to do a good deblurring when we know well the blurring function. In the case of this, it was simple. Get a distant star that doesn't have anything around, is a point, and look how that point gets translated into the telescope. Well, we didn't have something like that, so we did the following approach. We consider that the local resolution was our blur, and if we had a worse local resolution, we had a larger blur, isotropic blur. If we had a better local resolution, we have a sharper blur. And then with that local resolution blurring approach, we went into an iterative binary approach, nothing special. and we applied to a number of maps. And this could be considered like sharpening, but sharpening with additional information. I mean, the information about how your structure was blurred, how your structure was degraded to start with, or at least an approximation to how your structure was degraded to start with. And well, you have here the same channel TRPV1 that it was before. It was sharpened with a number of algorithms, and we made many measurements of quality, and one of them that you have here is the EM ringer. But really, the slide that I like the most is this one, in which you see how there are areas that normally correspond to loops in the structure. This is in the proteasome structure, that really are not very well seen. They are very unstructured, very disconnected when we apply other methods, and really when we apply one that takes into account some, if you want, prior of what has happened, that is actually an approximation to the blurring, well, this gets much better. This loop gets much better defined. And when we check actually at the level of the spectra of these maps, what is happening, well, what we find in most approaches is that there is a tendency to put the this is actually the broken line is the theoretically calculated spectra of that macromolecule, and what we see is that this B factor general boosting of high frequencies that is typical of many methods. What we see in our method, local deep blur, is a spectra that it fits much more to the one that has been, that would be considered the good one. I mean, is the one that has been theoretically calculated from the structural maps. The map could be better or worse done, and the way to calculate the map from the model always can be done better. But in principle, you see that the difference between this way of approach and this way of approach, or this way, is really very large. You may also know log scale, which is a very nice method if you have a map that is similar to your existing map. So you are solving a problem for which similar problems have been solved before. In that particular case, you have a very clear prior, is that whatever you want to reconstruct is similar to that other thing. And that method is also working very well at the level of the sharpening, and is working very well at the level of the spectra of the macromolecule. But the surprising or the nice thing is that we are putting much less information and much more general information. We are not asking for any map similar to have been resolved before. For any map, we can calculate the local resolution, and from there we can proceed ahead. And this, in this particular case, had some, in that collaboration with the GPCR, one of the many GPCR published in the last 1 year and a half by Cryo-EM, had some influence in the tracing that in turn had influence in how the pharmacology was understood. So that is local resolution, the directional local resolution, as a new tool that has, I think, a very broad applicability. How local resolution, or better still, in a short time, directional local resolution can be used to get better maps in a more focused and with putting more information, sharpening of the map. But there was something that we didn't like, to be honest, in the calculation of the local resolution as we were all doing it, is that at the end of the day, ResMap, monoRes, blockRes, they were doing some sort of signal-to-noise ratio. And we were then, especially when we were correcting in a global manner by the B factor, we were multiplying the signal and the noise by the same thing. So we were having the same coefficient, I mean, the same coefficient, no, the same division, the same value of the division of signal versus noise. However, your eyes were telling you that one map was quite different from the other, that one map was really much sharper than the other. However, your measure of quality was totally insensitive to that change. We didn't like that thing. We wanted to have some measure of quality that would be able to follow these enhancements that you thought you were doing to the map. And this was not so easy to find a way around. This is, by the way, a review we did on resolution in general. But how did we thought about that? Well, we thought in a new way. What about using deep neural networks here? And to do something that was very, very simple. Go to the PDB. The PDB, if I'm not wrong, is 140,000 structures, something like that. Okay, go to the PDB. From each of the structural models, create a map, and now low-pass filter the map by a different resolution, create many small boxes or small cubes, and present those small cubes to the network. And I am asking the network something very simple. When I present you a cube of something like that, please give me only one number, resolution. And that again, resolution, quality, blurring, whatever we call it, but something that really understand that it has features corresponding to the shape at that maximum frequency, or of a signal that has been filtered to that maximum frequency. So with that, if we now call it resolution by deep learning resolution, and we color the same GPCR that we solved or we applied monoRes before, well, we see a very similar result, and we see that really there are areas that are better resolved, there are areas that have a lower resolution, that are the same areas that we found by monoRes, ResMap, BlockRes, all of them, and we were happy. I mean, things is working in situation that they should be working. In fact, we went to study a number of sharpened maps, and we found what we expected. We found that all methods were giving you about the same result. So if for this particular virus, ResMap, monoRes, they were telling you something around 4. Okay, something the same for other structure at a low resolution. And this was for sharpened maps. So for maps that really I have done some B-factor enhancement. Now, what should we expect if instead of having sharpened maps, I work with unsharpened maps? Well, by my eyes, this map would be more blurred. However, ResMap, monoRes, BlockRes, eventually, would be telling me the same measurement. Since I am seeing them more blurred, I am expecting that my deep learning approach would also tell me that they are more blurred. And this is exactly what happened. So I'm sharpened, so more blurred maps. I have the same thing that before doing the sharpening. However, the deep res is telling me that this is much blurred than what I got from these other measures. Because these measures, they are insensitive to a boosting of high frequencies by a global B factor. So I have, and however, my deep red is not insensitive. I mean, when I do a sharpening, I get the same result that I got more or less with the other. So I have a very nice tool that is able to follow the enhancement. If I have a sharper map, I have a higher value of deep res. If I have an unsharpened map, I have a lower value of deep res. In spite, and so I don't have the problem of having the same measurement, however, seeing different things in the map. This also gave us the idea. Really, this was at the review process, so it is something very, very new. It gave us the idea that maybe this could be used to check on over sharpening. In the following idea, this is the block res or mono res or res map of a certain map. It's like that. No matter which boosting I do, which general boosting I do. Okay, I start now performing different boosting and checking with deep res. And I see how deep res is moving to the higher resolution as I increase more and more the high frequencies, to the point in which I surpass, I actually get a higher resolution by the deep res than with the other method. And at that point, we thought, well, listen, this could be one of the many ways to check that I am doing some over sharpening. This should not happen. Okay, so this is the story I wanted to tell you. It's really about local resolution, directionality in local resolution, how local resolution can be used to increase the quality, and how we wanted a local resolution measure that was able to follow what your eyes saw. That if you had a crispier map, it produced a higher, a better value than if I had a more blurred map. And this is, well, we are starting to extend the whole thing to tomography. This is for another time, and probably we are starting a new generation of local resolution method. I mean, the first generation, the block res, res map, mono res, I think have made a good job, but now we have to introduce directionality. We have to introduce a key question that I suppose you have made yourself more than once, that is local. How local is local? And it's not that easy. I mean, you cannot work reciprocal space and time space, I mean, or spatial are very much mixed. You cannot really have the best in the at the same time. So really, I'm sure that we are going toward a new generation of local resolution measures that will take into account both directionality and will explicitly address the notion of how local is local. And it's a point that we try to make everything simple to use. So our traditional software is XMIP, that is within a framework that is very easy to use. So easy that even for certain functionalities can be used over the web. And if you go to the EMDB in Europe, you go to validation tools, you get to that page, where essentially you can calculate the local resolution by whatever way you want to do it. And it's not yet included, but we would be including there the directionality and the deep res approach as well. So it would be a very simple way. I mean, the software works over the cloud and all that, so it's very simple to use. It's simple to install. but this doesn't need an installation for for this part that is over the web. Always looking for good people, and this is actually the team. Jose Luis is the person that has worked more on the directional local resolution. Carlos Oscar is the other co-PI of the laboratory that is here. Ernei, who is not in that picture, that this was taken in Christmas, and that was a time that he was not in the lab, he was on vacation. But basically, I have been talking about Carlos, Jose Luis, and Ernei mostly. And we are working a lot in collaboration with Heman Tagare, and we really want to. I mean, competition is fantastic, but we want to have a good set of mind working in a defined problem and trying to get the best possible solution. So this is all, and thank you very much, really.